All right. Thank you, everyone, for being here at our April Global Perspectives on Medicine, Rehabilitation, and Robotics webinar series. Today, we have a wonderful presentation on at-home stroke neurorehabilitation, the design and validation of a low-cost mobile EEG-based brain-computer interface. Our speaker today is José, or as he goes by, Pepe Contreras-Vidal, who is the Hugh Roy and Lily Kranz Cullen Distinguished Professor. He's a Center Director of the NSF IUCRC Brain, co-I of Music Dementia Research Network, the Director of the NSF IUCRC Brain REU and REM sites, and the Director of the NIH R25 Neuromotor Training Program. He's also the director of the Non-Invasive Brain-Machine Interface Systems Lab and in the Department of Electrical and Computer Engineering at the University of Houston. That's a lot of credits, but a little bit about him. He is an elected fellow of the IEEE and the AIMBE for his pioneering contributions to development of brain-machine interfaces, for controlling wearable exoskeletons for rehabilitation, and for mapping art-evoked brain activity. He led the first comprehensive knowledge integration assessment of the global state of clinical trials of implantable brain-computer interfaces, published in Nature Reviews Bioengineering. His work at the nexus of art and science is opening new windows to study the neural basis of human creativity, while advancing the understanding of the arts as an active neuromodulator of brain function with applications to the development of personalized art prescriptions. His art science team received the Chamber Music America's 2025 Interdisciplinary Collaboration of the Year for Meeting of the Minds, which was performed at the United Nations 2024 AI for Good Summit in Geneva in Switzerland. Dr. Contreras-Vidal is a member of the National Advisory Board for Medical Rehabilitation Research at the U.S. National Institutes of Health, and his career development in biomedical engineering was highlighted by the journal Science. So we're very excited to have him here today to present to us. And Dr. Contreras-Vidal, I'm going to turn it over to you.
Thank you, Rasha. It's a pleasure to be here today. And let me add the video here. So I need to stop this and do it again. The sound. Yeah. Okay, can you see my screen now?
Yes.
We go to presentation mode.
Perfect. Yes.
Great. Well, thank you. Thank you. Good morning, everyone. It's a pleasure again to share some of the work that I'm conducting here at the University of Houston Brain Center. And I'd like to invite people to, I don't know how we are answering questions, but feel free to, to ask any question anytime. And maybe Rochelle can help me to the logistics for that. Well, so first of all, just some acknowledgement in terms of support. I have industry support to the Brain Center, NIH, NSF, Mission Connect, and the IEEE. And so this is the outline for my presentation today. So I'm going to start with reviewing some of the work on brain-computer interface or brain-machine interface that led then to the development of we believe one of the first, or if not the first, accessible non-invasive BCI system for use at home. And I'm gonna discuss some of the challenges along the way, the state of clinical trials for this technology, some of the opportunities, and how we can maybe work together to make it more available. And time permitting, I'm going to touch on a new application for BCIs as in the context of art medicine or music medicine or dance medicine, which is something that we are working really hard to bring to treatment of mental health and neurological disorders and other conditions. Okay, so let's start. This is my favorite definition of brain-computer interface, and there are many. Unfortunately, lack of standards in this field has been problematic. This is from Jonathan Wolpatt, 1991. The brain-computer interface is an additional communication channel of the brain with the world using non-normal output pathways of peripheral nerves and muscles. What I like to highlight here, additional communication channel of the brain with the world, right? And if we add to that system BCI connectivity, thinking about Internet of Things, for example, then we have BCIoT, which allows for thought control of digital and physical environments. Now, this is a very loaded term, and it's really connecting our brain directly with visual and physical worlds, right? So that has many implications, not only in medicine, but how we interact in the classroom, how we interact at work, how we interact with others. So it's very important. Now, this started actually, this is about 50 years old now. It's a brain-computer interface architecture at UCLA by Dr. Vidal. And this would take a whole room, right? Now it's just a headset, but you can see on the left column, you see the user with, in this case, a brain cap, EEG, right? And some manual control, so on display. So typically, this system's feedback is through visual information conveyed to the monitor or TV, right? Then we have the control area where we get recording, printing, control the display, and then we have the computer area where we have the all the analog digital conversion and the actual decoding and all that, right? So this took a lot of effort back in 1973, but that's one of the first papers addressing this. Now, you may know that there's actually a BCI, non-invasive BCI, that has been approved already by the FDA, and this is from Neuroscience. There is a product code for that, QOL, which I thought initially was quality of living or life, but it's actually a new code for an EEG-based BCI. And I want you to, and this is taken from the 510K from the FDA when they approved the device. They mentioned EEG-driven upper extremity exerciser, right? It's a non-invasive prescription device intended for rehabilitation by driving movement or exercise of an impaired upper extremity in response to the detection of purpose-oriented electrical activity produced by the patient's brain. And the key word here is purpose-oriented electrical activity produced by the patient's brain, because that's really, we are talking about the intent, detection of the patient's intent for movement, which is basically what the BCI is being used for. And of course, once you detect the intent, you can use that to control external devices or visual worlds, right? So this is important because it means that there's already a code, that there's a device approved. So this can be used as a predicate for any new type of system that also fits the definition. And this is important, for increased accessibility and actually perhaps it's even speeding of the process, the regulatory process. Now, we conducted a study on the implantable brain-computer interface, right? And I'm going to go back to the non-invasive, which is what I focus, but I think it's very important to provide the global context here, right? And I conducted this together with my former student, Michelle Patrick, who is now at UT Health, and Ian Burhart, who is the founder of BCI Foundation. He was actually implanted for about 7.5 years. And so it was very important for us to include people with lived experiences as we were working on this review. And the main message here is that there are no implantable BCIs approved by regulatory agencies for the medical device market, despite being in clinical trials in 1998. And there's very little information about their progress and outcomes And I will refer you to this review where we address all these issues. I'm gonna just touch on a few points because I really want to go back to the work that we do here, but I think it's important to provide the context. So just to go a little more on the definition of brain-computer interfaces, right? And this could apply also to not only to implantable but also non-invasive, right? So we use particular sensor to capture brain activity, to record brain activity from the brain. These are all examples, 4 examples of implantable electro arrays, microelectro array. We have a neurotrophic cone, electrocorticography, to see the grid, or even endovascular array, right? And of course, they acquire information that needs to be transformed into a digital form for pre-processing and decoding, and then the output of these decoders can be used for communication or motor control. And there are many applications. These are some of them, and that are used to restore communication or motor control, right? And there are different types of feedback used to close the loop: auditory, visual, tactile. And some of the prosthetic devices have also sensors that provide biomimetic feedback, which then can be used to some pre-processing and decoding to microstimulate cortex to generate the percept of tactile feedback in this case, right? So it's an output brain-computer interface to the world, but also an input brain-computer interface to generate these percepts of, in this case, tactile feedback. And you can see on the picture on the right, our collaborator, co-author Ian, who was implanted for about 7.5 years with a BCI that was used in this particular case to control functional electrical stimulation of his right arm so that he could regain grasping, right? So this is a typical loop for brain-computer interface. Now, this is a recent plot that the MIT Technology Review based on our data. And I want to bring, so far, this is up to the end of 2024, there are about 38 people that are implanted, right? And about 28 or so clinical trials worldwide, right? So this is not a very large number. People have been implanted for 7, 8 years, perhaps, a few of them. And the representation, many of these are in the US, a few in Europe, China, Switzerland, right, also in Europe. And so there's not really a global concerted effort at this point. We have been working in silos, unfortunately. And this is just a brief summary from the paper. I just want to bring some, I think, important points. So we look at the at the groups here. The first column are the different groups that are doing this type of work. And then we have the number of people that they have implanted, right? And then the type of electrode, the type of BCI, input or output for communication, the sex and the age, right? And what we can see is that, in the early days, a few groups were working on this. They started Georgia Tech, and then BrainGate came. And what this grid tells you is from data available from many sources is, for each participant, the implantation date, to the best of our knowledge, and the the completion of the study or the removal of the electrode for any reason, right? So, and this, the details on the paper, but you can see that the longest implant, longest lasting implant was actually in the early era. And then with funding from the Brain Initiative, there was an explosion in the number of groups working on BCI, which remains up to today, right? So we found some challenges, right? For example, women are underrepresented in clinical trials, even when you account for differences in disease-based or injury-based prevalence, right? You can see here, up to the day of this review, 49 males against 11 females with also distribution in terms of the age of an equal distribution of the age of the participants. And so if you look in the range 22 to 45, there were only 26 men and only one woman, right? And so, this is something that we need to address in the field. Now, the other challenge that we found is in terms of data sharing and data ownership, right? So a total of about 2380 months of data have been collected as of December of 2023, right? Certainly there are more now. And with BrainGate having the largest number of participants, 16, with a neuroport array across 2 decades, right? Which accounts for 21% of the data. Of course, there are some concerns for patient privacy and data misuse, which limits data sharing. We noticed that only 39% of all the IBCI publications that we found that report participant data include a data sharing statement, right? And only a third of those provide a direct link to the data. So it's very hard to get these. We also found that data sharing statement do not mean that data will be shared. And actually, my group, tried to help with the analysis, and we couldn't get access to the data, even though we would be happy to provide data sharing agreement, right, and following all the guidelines. Of course, data sharing must be balanced against privacy considerations, right? And there are not many participants in IBCI, and many of them are on the news, so it's make them personally identifiable, right? So it's a really difficult. It's a challenge. The other aspect is, of course, the access to these systems, right? So currently, implantable BCI systems are limited to only a few patients with paralysis, tetraplegia, or dysarthria, who live nearby the PIs, right? There's lack of access also after the end of the clinical trial due to regulatory aspects, the need to maintain function and support, the need to provide system maintenance and repair for replacement, lack of grant or private funding. The PIs move, they relocate. There are legal aspects, so it's really hard right now. It's really hard to provide lifelong support. And of course, NIH and other groups are trying to address that, but this is really a challenge. Now, going back to invasive, non-invasive, this is from my lab, a demonstration of brain control prosthetic hand by a double amputee, a limb amputee. And so this goes back to 2016, but just to show that even with non-invasive method, in this case EEG, it is possible to extract information about intent and use that information to control prosthetic hands or other devices that they can use to restore function. Now, just to tell you a bit about the recipe, how we do that. So back in 2010, we discovered that in the space of electroencephalography or EEG, movement intent could be decoded from the fluctuations in the amplitude of movement-related cortical potentials in the delta band, very low rhythms, right? So this was amplitude modulation, or AM. A lot of people were looking at, features in the frequency domain and some other spaces, but it was really AM, and it was in the low frequency band known as delta, right? So the other important aspect of the algorithm is that you really need to look at the whole head, and most work use very low density EEG systems, right? You really have to have full coverage to be able to get this information, because you need to integrate across the space, the electrode, and also the recent paths. And the two important aspects is that you need to calibrate these systems, right? Because every brain is different, particularly when you have injury, and so to be able to obtain context, so that your decoder understand, how the environment affects your brain activity, you need to calibrate, preferably based on data from multiple sessions, right? Rather than just one day, right? So you really need to think about longitudinal studies, and many of the studies are just one single session, right? So it's very hard to generalize. And based on that, we show in 2010 that you could decode 3D center-out hand movements from EEG, which opened a door to basically develop the technology. Now there are some issues that we had to address, right? Which is these EEG signals are contaminated by many factors: temperature, humidity, movement, eye blinks, muscle activity, you name it, right? And I just want to show two examples, because I want to focus more on the global and aspects. But here, you have on the mirror plot a representation on the scalp of how the 15 blinks, a series of 15 blinks by the participant, will look like when you represent that activity on the scalp, right? So most of it is in the front, right? Here you can see the changes in EEG map in space, right? So they are in front, the eye generators are the front, and this is what happens on the last 15 blinks of a station of recording. So very similar. Of course, this contaminates the brain signal, right? So we want to remove them. The other point I want to make on the right is, so if you look at the amplitude in microvolts of the sensors across a midline on the scalp, so from the vertical electrooculography sensor here on the top left, and you move on the diagonal to these different sensors all the way to the back to PO6 on the back, you can see that the amplitude is changing, and in fact, it changes polarity around FC1. The other, and the shape as well changes. The other thing is that if you look at the impedance of the electrode, so what's the quality of the recording measuring on kilo ohms on the right, you see that the impedance measured at the beginning, at the end of recording changes, right? And so these are drift in the potential that if you don't take into account, it's going to affect your decoding. And this is something that, as far as I know, is disregarded widely in the literature, right? But with our adaptive filter algorithm, we can track these changes and account for them. And in fact, we have a very good estimate of the volume conduction of these brain signals as they propagate through the medium of the head, right? So if we take into account these factors, we can remove every sample the contribution of different type of artifacts to obtain clean brain signal that then we can process further for a BCI application. Now, the other concern that I ask very often is motion artifacts, right? And this is a paper we published on neuroengineering. We place an inertial measurement unit to track head motion, but also we were tracking optically the movement of individual electrodes on the head on the top left plot picture, right? And as people were walking at different speeds, in this case, I'm just showing 2 miles per hour and 4 miles per hour. They were walking on the treadmill. Now, if you look at the 2 miles per hour, before removing the artifact, we see activity related to the speed of gait. And you can see that on the lower plot, these peaks here that were related to the motion. And in fact, you see harmonics, right? And then after using an extended version of algorithm, we are able to remove these artifacts. And these are more clear when you walk faster. In this case, you have this motion artifact introduced by the walking. You can see the fundamental frequency and also the harmonic contamination, which is able to be removed after using our algorithm. And this is done in real time, which is essential for a BCI application. I will refer you to this paper if you are more interested on the technical aspects of this. So we can deal with artifacts. So this is an extension of the upper limb control by a BCI to the control of a walking exoskeleton. Our participant here has a spinal cord injury and doesn't have any sensation, clinically complete, no sensation, no control of his legs. And with the BCI, it is able to control walking and stopping on the exoskeleton. But these devices are very expensive. An exoskeleton like this, they go from 100,000, 150,000, which is really not accessible, right? And then there is also a lack of technologies like exoskeleton in children. So we have developed our own exoskeleton for children, and we are conducting a clinical trial for children with cerebral palsy. And so we're very excited about this, and we are also connecting their brain with the machine here. We think that promotes plasticity, and also it can have a benefit in terms of the sensory motor development of the kid. So we're talking about rehabilitation here. So we expect changes in the brain, right? So this is an example of that. Here we have 2 participants. On the left is an intact, able-bodied person. On the right, we have a participant with the spinal cord injury. And what you see on the skull maps are the relevant areas for the coding gate on these 2 participants. So we had 9 sessions or BMI training over a period of a month. And what you can see on the top left is that the representation in EEG space was changing over the first 5 sessions, right? As they were learning how to control the exoskeleton through the BCI, right? But then after 5 sessions, the representation, the areas of the brain relevant for the coding gate stabilize. They become very consistent and remain there, right? And you can see on the lower left is that there was improvement with session in terms of the overall accuracy of control with the BCI, meaning that the brain was changing as it was learning the internal model of the robot and the BCI. The decoder was fixed, so we didn't change anything in terms of the algorithm. So this was due only to the brain. Now, a similar pattern is seen with the participant with spinal cord injury. However, the actual relevance of the EEG sensor was different, right? So you can see that also it took about 5 sessions for this participant to learn, right, how to control the exoskeleton with the BCI. But the representation that the patient was using, the participant was using, is completely different from the able body control, right? So in the able body control, this is aligns with our understanding of the neural representation for gait in the brain, while certainly the participant with spinal cord injury was using a different network. And his performance, by the way, was better than on the able body participant, because they didn't fight the robot because they don't have sensation. So if you were to wear one of these robots, you will fight the robot because it doesn't feel natural to you. These are just approximations to our lower limb extremities. Now, so let me go to the neuroexon, which is the main topic today. Now, so we you will partner with colleagues at Till Memorial Hermann in the Department of PMR, and for the exoskeleton, we are a colleague at Rice University who had developed an upper limb exoskeleton, and we were in charge of developing the brain-computer interface or brain-machine interface. And so basically we had, people with, chronic upper limb impairment after stroke that had problems, controlling movement. And they went through 4 weeks of brain exoskeleton therapy in a very simple task, a target hitting task, just, elbow flexion and extension, very simple. But the exoskeleton was controlled through the BCI. So basically, it's a brain switch, right? If we detect intent from the participant, that will enable, the assistance needed exoskeleton to help the participant move his or her arm, right? And again, this was based on movement related cortical potential in the delta band, looking at the amplitudes. And what we found is that after the 4 weeks, the participant got better, right? And so the clinical protocol, this is published in our Image Clinical. So we have a baseline where, we tested clinically the participants 30 days apart, and to make sure that there was no spontaneous recovery. And then we went through a series of sessions to obtain data for the calibration of the brain-computer interface. And after that, the people enrolled in the trial went to 12 sessions of therapy with the BCI. And then we tested post-treatment at 2 weeks and 2 months. So that's what we did. And this is just to show you the cohort. It's a small cohort of 10 people. We screened about 120 people to get this stem because, it requires commitment to come for all these sessions. And if you live in Houston, you will know that traffic is really bad. and there were many factors that we tried to account for, in addition to the clinical ones. And so we characterized them, our clinical partners did. And as outcome measures, we have the Fogel-Meyer upper extremity test, the Action Research Arm test, and we also use Jepson-Taylor hand function test and the NIH stroke scale. And we had 3 participants that were severe to moderate, and the other 7 were moderate to mild participants. Just to go straight to the outcome, right? So this is group-based analysis. It revealed an improvement in arm and hand function, and at post in 2 weeks and 2 months, and they reached the minimal clinical relevant difference, and so we were very happy, and we had participants asking to keep the training because they had benefits on this. Now, we look at the changes in these MRCPs because, again, the decoder was fixed. So the change, the improvement had to be because the brain was learning. So this BCI was promoting learning. And in fact, we found a linear relation between the change in the amplitude of these MRCPs, pre and post. In this case, I'm showing with respect to the ARAT, right? So, and we can see here examples of the MRCPs at the start and end of the therapy, showing the nature of the change for this electrode. In this case, we have full coverage, so we look at all the electrodes. And so that suggested to us that this BCI was effective, right? So motor intention-based arm training targets activity dependent on plasticity. That's what we show. And these are very simple BCI. It's really a brain switch, right? Detecting intent. A majority of the participants recovered clinical relevant functional movements. By looking at the nature of the changes, the location, we found that ipsilational delta band EEG activity was highly correlated with motor recovery, and that suggesting higher activation of ipsilational hemisphere post-intervention. Now, of course, that was good news, but there were some drawbacks, right? Because the intervention was conducted at the clinic with a full engineering and PMR staff. The equipment is very expensive with a one-of-a-kind robot exoskeletons, talking about now $500,000 combined, right? And also the usability and the BCI performance of the device for use at home settings, was unknown. So it was clear that in order to increase access to this type of technology and also adoption by the end users, we need to develop a lower cost, secure, user-friendly, risk-free device for use at home, right? And so I'd like to spend some time and tell you what we had done to get here. And of course, it has to be user-centered design. So we started with our end users. We did interviews with the focus groups. We talked to not only to the participants, but PMR personnel, the physical therapists, occupational therapists, to make sure that we have all the input. Of course, there were several factors that we had to address simultaneously, right? One is interoperability, because we wanted our BCI to be able to communicate with different devices. Maybe off-the-shelf devices that you already use at the clinic. And we were very mindful of the cost to increase access, portability, usability to increase adoption, the form factor, the reliability of the system for use at home, and of course thinking also about the regulatory path to get this approved as a medical device. And so we combine the design of a brain-computer interface using custom amplifiers, dry electrodes that don't require gel or liquid, saline liquid, and also the software that remove artifacts and how the machine learning required to detect intent. And of course, an IoT module to be able to communicate with the external world, in this case, rehabilitation hardware. So this were part of the product specification for the device. And this is the device, the first iteration of the device that we tested, and we reported in the journal Sensor, right? So you can see that in this case, we went from full coverage. We learned that we didn't need all the electrodes. We only needed 5 over sensor motor areas, as well as eye sensors or EEG sensors that were monitoring the eye blinks, the eye movements. And on the back, we had the motion sensor, we have machine learning, the battery, communications, everything. So this is a self-contained brain-computer interface that can talk to, in this case, to this robot. There's also a graphics user interface that provides instructions to the participant, right? So this is a general purpose brain-computer interface with connectivity, right? And so this was the protocol for the proof of concept validation of the system, which we repeated based on the original study in neuroimage. In this case, after informed consent and baseline clinical assessments, right? We had training at the clinic up to 6 sessions to train the user to use the device by himself, and also to calibrate the decoder so that it could detect intent. And if the participant was able to use the device by himself and we were able to calibrate the system, right, then they will proceed to the at-home training. And in this case, they did 60 sessions, AM and PM, 5 days a week for 6 weeks. And then we had 2 post-clinical assessments right after and then 1 month after. So following the original protocol. And so, we follow an iterative BCI design, looking at the journey of the user in using the device. And these devices were tested not only in Texas, in Houston, Texas, but we have a participant in California and one in Maryland that took the device with them, right? And you can see here, for example, we found in 3 users early on that they had problems pairing the brain-computer interface with the robotic arm. That required an upgrade in the firmware to solve the problem. One of the participants had issues with inserting the micro USB connector to charge the device, and so we had to redesign that and replace it with a USB-C port, which is good because that's becoming a standard. And we saw some issue with the endurance of the battery or maybe the server locking, and that required some software revision. And although we trained them to use the device in a particular manner, when they were at home, they were just not necessarily following the instructions, right? So we had to reinforce the device by analyzing the failures of the device, right? And so we had 3 participants that had that problem, right? So the version that we have now has benefited from the input from the users using the device at their home, right? And this is an example of a recording. This is the MRCP from a participant with chronic stroke. So the vertical dotted line represents the detected intent for movement. You can see how the negativity in this MRCP that leads to detection of intent, right? So the feedback was very positive, compliance was high, usability was high, and so we have a cost solution here. Now, why this is important, right? I talk about accessibility, but let's compare our system, and what you see here in this plot is an electrode, a dry electrode from our NeuroExo system over FZZ. And remember, we have 5, right? So basically, this sensor is recording the activity coming, from motor areas, and these signals, need to travel through different layers of the brain, right? So it has to cross, cerebrospinal fluid, has to cross, skull, scalp, hair, so the propagation of these signals reaching the electrode are distorted, right, by the medium and artifacts, right? So we get a very tiny signal in the order of microvolts that is heavily contaminated and distorted, and so our algorithms had to increase the signal to noise ratio, had to identify and remove artifacts, right? Now, the there's a great work being done by Synchron with the Central. That's an endovascular array. So it's placed closer to the brain, right? So there's also volume conduction, but they have fewer layers that they need to cross to get the signal, right? So you can compare here these layers versus all these paths that we need, that our signal need to travel to be detected by NeuroX, right? So so the bandwidth, the frequency range that the synchron electrode can see is probably twice as big as what we can see with EEG here, right? But we have more control on where we place the electrodes, and we can place several of them. They have less control, and it's still an implantable device. So there's some risk, even though stent technology has advanced quite a lot. Now, if we compare in terms of the technology, right? So we had the Synchron, we had the NeuroExo. We are recording EOG. They don't. They actually use eye tracking in many of their experiments, an external device. Record EEG, we have the National Measurement Unit for head motion, and we record impedance. They also record impedance. They have endovascular ECOG. They can also, although they haven't shown that, they claim that they can stimulate too. Anyway, what's interesting about this is that after the end, at the output of the decoder, we have machine learning or a classification layer, right? So we use support vector machines of LDA. We can actually place anything we want there just by replacing the software. They also use support vector machine. So what we have at the end is, in this case, we have a binary switch for intent. They have short clicks, long click, or no click, right? So it's very similar. We could probably replicate the same. And we had the communication through Wi-Fi and Bluetooth, right? So with this, we can control many types of external devices. I just show one for the exoskeleton. So they're very similar. It's just where the sensor is located, right? But they are both affected by volume conduction and artifacts. But I think we have a good solution here, and so we're very excited about it. Now, so I'd like to take just a couple of minutes to tell you about how we're using this technology now in the arts, right? And this is important because we know that music, for example, can trigger memories, can change your mood, right? And we have now, in some programs on music therapy, music neurological therapy, music in medicine, right? So I think it's very important to understand how we can personalize a potential arts prescription, be music or expressive movement, dance, or both together, right? So, and of course, to do that, people need to have access to the technology in different contexts. So this is a study that we did with a professional jazz musician wearing the brain caps, and basically we vary how they join the session, right? So initially we have one participant that started to play. There after 5 minutes we have the second one, and then the third one, and we change the order, we balance the order, right? And of course we had some baseline recordings to check the impedance, to look at eyes closed, eyes open, to understand baseline, right, before they went to play together, right? And these scale maps on the right, they represent the quality of the signal. So, like if you look at in the second row, mirror column, there are not those, meaning all the impedances were really, really, really good. We had excellent recordings. But in the other, we just have a few, right? And so this is very good, actually, because they're, even though they were moving and they were performing, they're really performing, we were able to get good data. And these are panel D just shows some examples of the activity that we recorded at the wear plane. Now, so just to give you a little idea about what they were playing, in this case, there were two people, the drummer, the sax player, and then on the third segment, all three were playing. So remember, they were improvising. This is jazz, and so because they were wearing caps, we synchronized all the recordings through a process known as hyperscanning. We are able to look not only at the interbrain synchrony, so how their brain areas talk to each other, but also interbrain synchrony, how the brain activities relate to each other. So now on the right plot here is a measure of interbrain synchrony, right? So if you remember, at the beginning, there's only one player, right? So in this case, P3 starts. So P3 starts. And when we look at the interbrain synchrony between P1 and P2, who are just listening, right? We see very little, right? But as soon as P1 joins and then P2 joins, when we are comparing P1 to P2, then we see an increase in interbrain synchrony for those musicians, right? And if we change the order, in this case, we have P1 to P3, right? So P3 starts here, and then P1 joins, and you can see an increase in the interbrain synchrony, which remains throughout the event. And lastly, we have the other combination, which is P2 to P3, right? That only happens also at the end, right? So this is, a way to look at the brain dynamics, not only within each brain, but also across the brain. This is very important for looking at social interaction and anticipation, turn-taking, teaming. I want to jump on this because I don't have too much time, but this graph summarizes the findings, right? So, and this is telling you, the interbrain synchrony, between participants in two frequency bands, in this case, beta and gamma. So when they were just passive listening, very little you could see. When one was active and the other passive, we started to see a network, right? And mainly on the posterior part of the head, right? It's purely sensory, right? Very little going to the frontal areas. But then when they both were active, then we were able to discern better, the network that was recruited as they were improvising together, right? We see more clearly, but not as a dominant, right, frontal involvement, but certainly it was very sensory, right? So the posterior part, they were looking at visual cues, they were looking at auditory cues, to help them improvise. So we are conducting this type of studies in different art domains, trying to get normative data so that we can figure out how we can then use AI to personalize sound or music to gain access to specific parts of the brain, right? So to conclude, these BCI systems are really powerful tools to understand the brain in action and in context in real-world settings at home or in the theater or at school, right? It's very important to quantify the coupled brain behavior, right? In this case, the brain activity as you undergo a music therapy session, for example, or in this case, as you were talking about performing, right? And so we believe that it's possible to use this technology to develop in the future arts prescription to treat, TBI, stress, Parkinson's, or maybe Alzheimer's disease, right? And the last point is that it has been really amazing, the response from the public. They enjoy this performance. They enjoy learning a bit about the brain and about the technology. So with that, thank you very much. I'm happy to answer any questions at this point.
Thank you, Jose, for that. We're running a little short on time, but I think we have time for a few questions. If you have any questions, you can put them in the chat or put them in the Q&A session. Actually, given probably most of the people on the call are living in a low and middle income country. And so some of the presentations in terms of the high robots may not necessarily be directly applicable. So I wanted you to take some time to just talk a little bit about the low cost BCI system. Maybe what is the cost? Is it available? Can it be, used now or purchased for experiment in low and middle income countries? And the cost to set up the type of experiment that you discussed. Say someone has a different robotic device or a different instrument that they want to test. Can you talk a little bit about that?
Yeah, not a great question. Thank you, Michelle. So, the BCI device has been designed to talk to any other device that's IoT enabled, right? That can communicate through Bluetooth or Wi-Fi, right? So poses no constraint on the other side, right? Which means you can use in different contexts, right? Now, so right now we are at the point that where to get regulatory approval as a medical device, we need to go through the FDA, and that's the next step for us, right? So it's not available yet in the market because it hasn't been approved. It's only a research device, right? And so under IRB, right? But I'm very happy to certainly to collaborate with people that might be interested in using this technology in some other places, right? Now, there has been designed, built, and tested in my lab by students, so it's actually very low cost, under $1,000. It's just economy of scale, right? So certainly to produce 1,000 of these tests, I can not do in the lab. I will need to send it to a company to build it, right?
Yeah.
But the whole idea is making something that's accessible, that can be adapted. There are many spin-offs that we are already investigating here, right? So and it can be used in children. It can be used, for delivering different type of intervention. So I'm happy to talk to people interested. And as we build the next batch, maybe we can include some others, or depending on the number of people that are interested. But this is very accessible, okay, compared to other technologies anyway.
Yeah, no, that's exciting to know that you would be welcoming research collaborations, et cetera. I mean, I might talk to you about bringing one of those to Botswana. I had a question about the frequency band.
Yeah.
So in the low-cost BCI system,
Yeah.
you mentioned that the delta band was where you saw correlations to improvement. Can you talk a little bit more about that? You saw, and then later on in the music work, you talked about, looking at the beta bands and the gamma band. So can you just maybe for the audience who might be more clinical, just kind of talk a little bit about why that might be the case?
Yeah. So first, more generally, right? So if you look at the EEG recordings, ECOG, local field potentials, right? What you see is, I think, is more like a U shape. That meaning there's information in the low frequency band like delta, but there's also information clearly in gamma, right? And mu, but it's like a U, right? So there are groups using gamma, right? The synchronous using beta, right? And we're using delta. So we have shown over and over, it has been confirmed by many groups that there's valuable information in the delta band, which has been thought to be just related to sleep. I mean, people didn't look at delta for volitional movements or of the body, right? Even speech, right? So it's surprising, but there's information there. It's the amplitude modulated, right? So now that doesn't mean that the other frequencies are not important, right? And in fact, we see the highest modulation when we are looking at interbrain synchrony in the gamma range, right? So it's more localized, right? Delta is more broad, right? It's more localized, it's lesser in amplitude, but it's very consistent, right? And we can see that how it's changing. We have a longitudinal study or several months of two dancers, together, and we saw a gradual increase in this gamma interbrain synchrony over this, right? Meaning, they were developing an internal model of each other and taking into account the choreography, the music, the stage, so that they were just it was automated by the end. They didn't need to think about the performing part, right? They were, it was just a sequence that was released. So this is very new findings. So I'm happy to share more. But yeah, so gamma is very important, and of course we can we can tune the system, the headset, to focus more on the gamma. That some type of artifacts are more prominent there, hard to get out, but I think we're gonna get there.
Excellent. And the last question is, some of your algorithms. So for people who might be doing some work in BCI,
Yeah.
you've come up with some really great algorithms for noise cancellation, for
Right.
dealing with head movements, etc. Are those open source, or are they available to people to use for their own work?
Of course, for non-commercial use,
Yeah.
they are published. We like to share everything because it is so important. So yeah, and I get students in my lab that get trained on this, and they go back to the lab and they implement. So I really believe on the open science framework, and so yeah, so this is all published in Journal of Neuroengineering, and we're extending the general purpose device. So I think it's a good way to deal with this artifact.
So should someone contact you directly if they wanted to use some of your algorithms for even, because it sounds like a lot of your these algorithms are usable with any EEG hat as
Oh
long as
yeah,
you
yeah,
have
it's
the
agnostic to your
information.
system. Yeah.
Yeah, so is that, do you have it on, GitHub, or should they contact you to reach out to get some of those
Yeah.
processing algorithms?
Yeah. I mean, it's published, and of course there's some math on that, but it's not particularly complicated, right? It's an adaptive algorithm, and it is both sample adaptive, and second is electrode based, right? So it doesn't matter how many channels you have, right? And see, the problem is that up to now, many people use template subtraction of an eye blank. But we just showed that that's changing over time. You can
Yeah.
just pretend that the artifact's always the same.
Mm-hmm.
But you're testing on the field, right? So yeah, happy to talk to anybody. I'm happy to host the students. We're gonna actually have we have workshops as
Okay.
part of our music and dimension network, where we're trying to make this technology more accessible to people that from therapy or medicine. So happy to talk to anybody. Yeah.
Yeah, can you you can drop your email in the chat, so if anyone is interested, can you drop it into
Yeah.
to everyone? So
Yeah.
anyone who's interested can grab. There is a question that was placed in the chat. One person asked, would you be able to shed some light on the regulatory landscape battles that may accompany these neurotechnologies in addition to possible reimbursement for insurance?
Yeah, so I say that for EEG based BCI, there's already an approved device, clear device by the FDA. So you have a new one, that could be a predicate which will facilitate, approval, right? For implantable, that's a difficult question, right? It would take me time to answer that. But reimbursement, the FDA is making a lot of efforts in having some sort of concurrent review, right? So that as they go to the safety and efficacy review, that CMS is also already looking at reimbursement, right? And so there is reimbursement for the Neurolutions device as well. So the future looks bright, but we need a solution that is lifelong, right? Particularly when you're talking about implantable device. So, and there's a lot more private investment, so I think that's already accelerating this. But look, we have only, less than 60 people.
Yeah, who's done it feels like the implantable stuff is not as easily transferable as the non-invasive things,
Exactly.
to different countries, because implantable requires then expertise in terms of neurosurgeons and things like that. While feels like even though it feels like your solution for the non-invasive is powerful because it's low cost and it doesn't require medical expertise to use it per se.
Right. Yeah. So we put a lot of effort on the manual, for example, right?
Yeah.
Because that's so important, right? And communicating with the user was really eye-opening for us, right? Because it's the BCI is really a social technological tool, and
Yeah.
we forget about the social aspect of it.
I love that. I think that's powerful, the work that you're doing. I know we're running out of time, and to respect people's time, we've been a little bit over. It's been fascinating to talk to you, Jose. It's just been cool, the work that you're doing. Hopefully I put his email in the chat. Probably some of the references that you gave in your paper can be found in your Google Scholar.
Yeah, everything is on my Google Scholar, or I'm on the different, things. We publish a lot of data on scientific data, it's data descriptors. We're trying to put effort, although, that costs money, right? So it's and also standards, right? So there's a lot of work. And yeah, feel free, anybody to reach out. There's a lot more information. You Google IUCRC Brain. So that's the whole center, right? So we work with many institutions and companies to facilitate this.
Awesome. Thank you again. And thanks everyone for attending. We will have another ICOR is happening. Will you be at ICOR Rehab Week, Jose?
No, unfortunately not this year, but yeah, that's a really good meeting. I recommend it. Yeah.
Yeah, so next week, on the third week in May, overlaps with the ICOR, the Rehab Week session in Chicago, and myself and Dr. Mendonça will be attending. So we will not have a May event, but we will put the links to a lot of the ICOR sessions. We'll, maybe provide, so I think it's largely in person, but those are kind of super relevant activities that would be relevant to this discussion. But we will be back in June, and we will see you guys then.
Yeah.
Take care, everyone. Bye-bye.
Thank you, Lisa. Thank you.